Hello, everyone. I'm glad to present today my research project, which is related with the production of extracellular membrane vesicles of Clostridium perfringens as a potential vaccine against necrotic enteritis. My name is Laura Guerrero, master student at the University of Montreal. As numerous publications have shown, most cells like eukaryotic, gram-negative, and gram-positive bacteria actively shoot nanosized structures into the extracellular environment, which are called extracellular vesicles. Various names have been used to describe them. For example, membrane vesicles for gram-positive bacteria, outer membrane vesicles from gram-negative bacteria, exosomes or ectosomes for mammalian cells. Furthermore, extracellular vesicles have been found in natural environments, in vitro experimental conditions, and tissues or biological fluids. In general, these conserved structures are spherical, with an average diameter of 10 to 400 nanometers, and they have a well-defined membrane. Also, they are harboring bioactive proteins as lipids, nucleic acids, and metabolites. It is important to mention that depending on the nature of these compounds, the functions between each vesicle may vary. Knowing that our research is based on Clostridium perfringens as a gram-positive bacteria, the causal agent of necrotic enteritis in chickens, it is important to focus on gram-positive extracellular vesicles. Here on the left side, we can see a diagram representing the process of extracellular vesicles biogenesis. Initially, we have the cell with a peptidolicane wall delimited by cytoplasmatic membrane. In many cases, there will be various factors which will trigger the outer membrane bubbling because of the destabilization of the peptidolicane wall and cytoplasmatic membrane. Some of the factors can be DNA damaging agents, signaling compounds, degrading enzymes, hydrophobic compounds, and antibiotics. A better knowledge of these trigger factors, it is important because to improve extracellular vesicles productions under different environmental conditions. After the membrane bubbling, we can see the EVS structures as a component. Here inside, we can see the components. This EV structure has multiple functions. Among the main ones, the capacity for material exchange by intracellular cargo delivery. Furthermore, vesicles also allow the survival of the bacteria, prevent the antibiotic degradation, and finally, they allow to competition through antimicrobial activity. The production of the membrane vesicles is based on 4 steps, mainly the growth of the bacteria in a nutritive culture media, following by a cycle and ultracentrifugation process. Analysis of the vesicle production is done by protein quantification, direct observation with electronic microscopy, and particle size and distribution analysis. Within the preliminary results, here we can see extracellular membrane vesicles from Clostridium perfringens. By using our protocol, we were able to produce membrane vesicles. Here we can see round structures with a delimited membrane. The size is approximately 200 nanometers, and knowing that Clostridium perfringens is able to produce membrane vesicles, we can now continue with our next step of our new project. To sum up, Clostridium perfringens is the main causal agent of necrotic enteritis in chickens, damaging the intestinal mucosa and causing diarrhea and mortality, which lead to economic losses. It is therefore important to find an alternative against the pathogen. We have demonstrated before that this bacteria is able to produce EV-like structures. Our next objective are to verify the EVS immunogenic activity and their ability to protect chickens against necrotic enteritis. Research with other pathogens has shown that EVS have an immunogenic activity. Thank you very much, and I'll be here for your questions.
Thank you very much, Laura, for your presentation. I have no question from attendees, but I do have a question for you. If you can come on screen, please. Oh, sorry, we don't hear you. Oh, we have, thank you. So I do have a question now from the attendees, which is the following. Hello, Laura. So the question is, gram-positive bacteria have no outer membranes. What is actually contained in the membrane vesicles you've mentioned?
So related with the question, like gram-positive bacteria have no outer membrane. So basically, the thing that you can find inside of membrane vesicles that I mentioned before is basically just lipids and protein, and genetic material is going to be inside of the membrane vesicles. That's going to be the mainly content that you can find inside of them.
But do you plan to do further analysis of the membrane content of the extracellular membrane content or vesicle?
Okay. Yes. Well, in the future, like we are going to start doing some analysis about the proteins, and we are going to start analyzing which proteins are going to be able to find it, and to like that we are going to be able to have like to do like a comparison between them. So actually the content that we are going to study is proteins. That's why we are going to establish our protocol to start with the next step.
Okay. How do you determine that how do you determine, there's another question here where the attendee is asking how you determine that what you're seeing are membrane vesicles. Do you have markers for this?
Well, for now, that's a good question, but for now, the only way that we can say that it's a membrane vesicles is when we are doing the visualization with the microscopy electronic. So there's going to be some colorants that are going to stain the membrane. So when you're going to do like the visualization, you're going to be like the iron structure that is going to delimitate all the circular structure. So that is, with that is like a good hint to say that we have already membrane vesicles. Until now, we don't have any specific marker to each of them, but that's why we need to try to find which is the content inside of them to establish more protocols that are going to be accurate for them.
And one last question before we move to the other speaker. How do you select the Clostridium perfringens strain used to generate the vesicles?
Well, we are already in our project. We decided to work with two strains, like a pathogen and an commensal strain, just to make a comparison. But until now we are just working with the pathogen strain. So we decided to start working with pathogen strain to know if the strain is going to be able to produce membrane vesicles. Actually, the strain is just a pathogen.
Okay, well, thank you very much, Laura, for your presentation.